Welcome, curious minds, to The Deep Dive. Ever thought about how tiny clues in DNA can unlock really big secrets, especially in fields like plant breeding? Well, today, that's exactly what we're diving into, the fascinating worlds of PCR-based molecular markers. Our mission is to break down a whole stack of detailed course material on this. We want to give you those aha moments about the tools scientists use to, well, read DNA variation. By the end of this, you'll have a really solid handle on three key marker types, RAPD, AFLP, and SSRs. We'll look at how they work, their pros, and yeah, their surprising cons too. Then we'll switch gears to the art and sometimes the headache of marker scoring. We'll go from simple diploids to the frankly mind-bending complexity of polyploids. Think of this as your shortcut to getting up to speed without getting totally lost in the jargon.
It's a really important area, and you have to remember, before PCR came along, looking at DNA variation was much harder.
Right.
You needed quite large amounts of DNA, and you were mostly relying on restriction enzymes alone, the ones that cut DNA at specific spots. PCR polymerase chain reaction just completely changed the game. It lets you amplify tiny bits of DNA, specific regions from really small samples. It opened up genetics in a massive way. Much more efficient, much more accessible, truly revolutionary.
Yeah, that really sets the stage.
Yeah.
So let's jump into our first marker type, an early one, RAPD. Random Amplified Polymorphism DNA. This was one of the first techniques that allowed for, medium throughput analysis, right? Especially big in plant breeding early on. The idea seems almost deceptively simple. Using these incredibly short random primers, like 10 or 12 nucleotides?
Exactly, just 10, sometimes 12. And the random part is key. They're designed arbitrarily, so theoretically, they could land almost anywhere on the genome. During the PCR, if two of these primers happen to bind close enough on opposite strands pointing towards each other, boom, you amplify the DNA between them. You see that as a band on a gel. The polymorphism, the difference between individuals, often shows up when a band is missing. That usually happens because of a tiny change, like a single nucleotide polymorphism, an SMP, or maybe an insertion or deletion right where a primer would bind.
And you mentioned that the 3' end of that primer binding site is absolutely critical for this whole thing to work. Why specifically that end?
Good question. It's all about the DNA polymerase, the enzyme that actually builds the new DNA strand. Think of it like needing the perfect key in an ignition. The polymerase absolutely needs a perfectly matched 3' end on that primer to grab onto and start adding new nucleotides. If there's even a single base mismatch right there at that crucial starting point, the polymerase it can't start. No extension, no amplification, no band. So yeah, a tiny typo in the DNA can completely prevent that band from appearing.
So the absence tells you as much as the presence, in a way. Okay, so RAPD had some clear advantages back then. Simple setup, right? Just PCR and a gel. And crucially, no need for prior sequence info. That must have been huge for species nobody had sequenced yet, these orphan crops you hear about.
Absolutely. It really democratized things. Let labs work on less studied species without needing a massive sequencing project first.
And it was medium throughput. You could get, 2 to 5 different genetic markers, loci, segregating in one go?
Yeah, roughly 2 to 5 per reaction, which wasn't bad for the time. It was a definite step up.
But, and it's a big but, this is where RAPD ran into serious trouble, wasn't it? The Achilles heel.
Oh, absolutely. The major drawback was poor reproducibility. Big problem. Those short primers, they weren't very specific. They could bind inconsistently. Tiny changes in your DNA quality, different batches of reagents, even the brand of polymerase. All of that could make bands appear or disappear between runs. It was really difficult to compare results reliably, especially between different labs.
Wow, that sounds incredibly frustrating. How did researchers even cope with that?
Well, you had to be meticulous with controls. Using positive controls was standard practice samples you knew should give certain bands. You'd run them every single time. If the control looked right, you had more confidence in your other samples for that specific run. And yes, people would sometimes stick to the exact same brand, even the same lot number of crucial reagents like Taq polymerase for an entire project, just to minimize variability. It really highlights how sensitive it was.
Okay, so clearly that level of, let's call it controlled frustration, wasn't sustainable. Scientists needed something better, more reliable. And that leads us to AFLP, Amplified Fragment Length Polymorphism. You called it a hybrid, building on older methods but adding PCR power.
That's a great way to put it. AFLP cleverly mixes restriction enzymes like the older RFLP method with PCR amplification. You start by cutting up the genomic DNA with 2 different restriction enzymes, usually one that cuts rarely, a rare cutter, and one that cuts much more often, a frequent cutter. This generates a whole mix of DNA fragments.
But if you've just cut the DNA, you don't know the sequences at the end. So how do you get PCR primers to stick?
Right, that's the clever bit. You ligate or attach these short synthetic DNA sequences called adapters onto the sticky ends left by the restriction enzymes. These adapters have known sequences. So now you do have known sequences at the ends of all those random fragments, perfect binding sites for your PCR primer.
Ah, okay. And then comes the selective amplification part. The primers match the adapter plus a little extra sequence at the end.
Exactly. The primers are designed to match the adapter sequence perfectly, but they also have a few extra nucleotides, maybe 2 or 3 right at their 3' end. These are the selective nucleotides. This adds a whole second layer of specificity.
So you're looking for differences in 2 places then?
Precisely. You get polymorphism if the original restriction site was missing, so the enzyme didn't cut there. Or you get polymorphism if the sequence right next to the adapter doesn't match those few selective nucleotides on the primer's 3' end. Again, that 3' end match is critical. No match, no amplification for that fragment. This dual check makes AFLP much more specific and generates a lot more markers per reaction.
So that extra specificity made AFLP much more reproducible than RAPD, which was a huge improvement. And like RAPD, still no need for prior sequencing. Still medium throughput. Sounds like a winner.
It solved a lot of problems, definitely. But there were still trade-offs. For one, it was still a dominant marker.
Okay, dominant marker again. Remind us that means you see a band, but you don't know the underlying genetics completely.
Exactly. Presence versus absence. If the band is there, the individual could be homozygous dominant, AA, or heterozygous AAA. Both look the same on the gel. You only know for sure if it's homozygous recessive, because the band is completely missing, so you lose that dosage information.
Right, and AFLP, it sounds more evolved than RAPD. More steps in the lab.
Oh, yeah. It was definitely more laborious and costly.
Yeah.
You had the DNA digestion step, then the adapter ligation, usually a pre-amplification step, and then the final selective amplification. More steps mean more enzymes, more reagents, more time. More potential places for things to go slightly wrong, too, though it's generally robust.
Was there any way to streamline that multi-step process?
Well, one practical aspect helped a bit. Once you did the initial digestion and ligation, that adapter ligated DNA could be stored. You could then use that same template DNA over and over again for different selective amplifications, trying out different primer combinations with different selective nucleotides, so you didn't have to repeat the entire process from scratch each time. That saves some effort.
Okay, so we've had RAPD, the simple but shaky pioneer, and AFLP, the more robust but laborious hybrid. Now, let's get to what many consider the real workhorse, especially later on. SSRs, microsatellites, simple sequence repeats.
Yeah, SSRs are fundamentally different. What's really cool about them is that they're regions in the genome made of short DNA sequences, just 1 to 6 bases long, repeated over and over again in tandem, like ACAC or GATA. The variation we look for here isn't just presence or absence of a band, it's the length of the fragment, which depends on the number of repeats. More repeats make a longer fragment, fewer repeats make a shorter one. And you detect this using specific primers designed to bind to the unique DNA sequences that flank or sit on either side of the repeat region. Those flanking regions are usually stable, conserved across individuals, but the repeat region in between, that can vary wildly in length. It's like a unique genetic fingerprint.
And how we actually see those length differences has evolved quite a bit too, hasn't it?
Oh, definitely. In the early days, you'd use standard agarose gels, maybe polyacrylamide gels with silver staining for better resolution. These could separate fragments based on size. But the modern standard, especially for things like forensics or paternity testing where precision is paramount, is capillary electrophoresis. You use fluorescently labeled SSR primers, and the machine detects the different sized fragments as they pass a laser detector. It gives incredibly high resolution, down to single base pair differences.
Now, there's this interesting phenomenon with SSRs called stutter. What exactly is that? Sounds like a glitch.
It kind of is, but it's a useful glitch. Stutter happens because DNA polymerase, the replication enzyme, sometimes struggles with these highly repetitive sequences. As it's copying the repeat region, it can sort of slip, either adding an extra repeat unit or missing one. The slippage creates minor DNA fragments that are 1 repeat unit shorter or longer than the main allele. On a gel or electropherogram, these show up as faint stutter bands or smaller peaks right next to the main true peak.
Okay, but why is that stuttering actually important?
Because that same polymerase slippage mechanism happening naturally in cells over time is thought to be a major reason why these microsatellite regions are so highly variable in the first place. That inherent sloppiness in replicating repeats drives the high mutation rate at these loci, constantly generating new length variations, which is precisely why they're such fantastic, informative markers for us. The bug becomes a feature.
Ah, that's brilliant. A biological imperfection turned tool. Now, SSRs sound great, but developing those specific flanking primers must have been the tricky part initially.
It was the main bottleneck, yes. High cost, needed prior sequence information for every single SSR locus you wanted to study. Back in the 2000s, a common approach was creating enriched SSR libraries. It was quite clever. You'd chop up the genome, then use probes, little bits of DNA designed to stick specifically to repeat sequences, often tagged with biotin to fish out only the fragments containing SSRs.
Like molecular fishing hooks grabbing just the repeats.
Exactly. Then you'd clone these captured SSR-rich fragments into bacterial plasmids, those little circular DNA carriers in bacteria. Once cloned, you could easily sequence the inserts to find the unique flanking regions on either side of the repeat. That gave you the sequences needed to design your specific primers. And another big win was transferability. Because those flanking regions are often conserved between closely related species, primers developed for, say, sour passion fruit might work just fine for sweet passion fruit. That seemed a lot of redundant effort.
So a big upfront investment in primer development, but the payoff seems substantial. What really makes SSR stand out?
The advantages are huge. First, and maybe most importantly, they are co-dominant. This is critical. Unlike the others, SSRs let you distinguish a homozygote, AA, one band size, from a heterozygote, AA, two different band sizes. You see both alleles. Second, they're often multi-allelic. At one single SSR locus in a population, you might find many different repeat lengths, not just two options. That's a lot more information per marker. They're also highly reproducible because the specific primers target unique spots. And you find SSRs scattered all across the genome, which is great for mapping genes or studying diversity.
So the downsides were mainly that initial development cost and, maybe historically, lower throughput analyzing one locus at a time, though capillary systems helped there.
That's basically it. The cost and effort to get started were significant, but the quality and type of data, especially the codominance, made them incredibly valuable for many research questions.
Okay, so we've generated all this data bands on gels, peaks from capillary machines. The next vital step is turning that raw output into something you can actually analyze. That's marker scoring.
Precisely. Getting the data is half the battle. Interpreting it and converting it into a numerical format for genetic analysis is the other half. Let's start with the simpler case. Scoring in diploids, organisms with two sets of chromosomes like us.
Right. For those dominant markers, RAPD and AFLP is pretty basic, but with that information loss we talked about, presence of a band is scored as 1, absence is 0. Simple. But the limitation remains. A 1 could be homozygous dominant AA or heterozygous AA. You only know for sure that 0 means homozygous recessive. In a standard F2 cross, you'd expect that 3-to-1 ratio of presence to absence reflecting this.
Correct. Now, switch to codominant markers like SSRs. Here, we get much richer information because we can see the different allele sizes. A homozygous individual, say AA or BB, shows one band size, but a heterozygote, AB, shows two distinct bands, one for each allele. This lets us score more informatively. Often you'll assign numerical scores like 0 and 1 or 2 sup based on the number of copies of a particular allele, maybe 0 for genotype AA, 1 for AB, and 2 for BB. This reflects the underlying genetics much better and leads to that classic 1.2.1 segregation ratio in an F2 population. More information captured.
Okay, are relatively straightforward. Now brace yourselves as we dive into the really complex world, scoring in polyploids. This is where things get messy.
They certainly do. And it's useful to quickly touch on the difference between the two main types of polyploids as it impacts scoring. You have autopolyploids and allopolyploids. Autopolyploids arise from genome duplication within a single species. Think of something like hexaploid sweet potato. 6 sets of chromosomes, all basically homologous, derived from one ancestral diploid. During meiosis, chromosome pairing can be very complex, leading to really complicated inheritance patterns. They're also often outcrossing.
Okay, and allopolyploids, they're different how?
Allopolyploids result from hybridization between different species, followed by chromosome doubling. The classic example is bread wheat. It's hexaploid, but it contains 3 distinct subgenoms, A, B, and D, from different wildgrass ancestors. Usually in allopolyploids, chromosomes show preferential pairing. The A chromosomes pair with other A's, B's with B's, and so on. They behave a bit more predictably in meiosis than autopolyploids, and they are often self-pollinating or autogamous.
So multiple chromosome sets, complex origins. This must create huge challenges for scoring markers, right? What are the main problems?
Oh, absolutely. First, the sheer number of possible genotypes just explodes. If a simple diploid cross gives you 3 genotypes in the F2, a cross between 2 tetraploids, 4 sets of chromosomes, can result in something like 36 possible genotypes. Trying to track Mendelian segregation becomes incredibly difficult.
Wow, 36 possibilities. Okay.
Yeah.
And then there's the issue of redundancy, especially with gel bands.
Exactly. This is a major headache, particularly for markers like SSRs visualized on gels. In a polyploid, say a tetraploid again, an individual might have 4 copies of a particular chromosome region. Several of those copies might carry the exact same SSR allele, the same length, the same band size. So you see a band on the gel, maybe it looks quite strong, but you just can't tell if it represents 1, 2, 3, or all 4 copies of that allele.
So you lose the ability to determine the dosage, how many copies of each allele are actually there.
Precisely. That inability to determine allele dosage is arguably the biggest limitation of using these traditional gel-based markers in polyploids. You might see 2 different bands indicating heterozygosity, but you don't know the ratio. Is it 3 copies of allele A and 1 of allele B, an allele, or 2 and 2, an ABB, or 1 and 3, ABBB? The gel band presence, absence, or size alone just doesn't give you that quantitative information. And dosage can be incredibly important for how genes function and affect traits. It's a huge piece of missing information.
Although, I think you mentioned there's a slight exception sometimes in allopolyploids.
Yes, occasionally you get lucky. If an SSR marker happens to amplify from only one of the subgenomes in an allopolyploid, and there's no equivalent allele, essentially a null allele, in the other subgenomes, then that marker might effectively behave like it's in a diploid. You might be able to score a 0, 1, 2 based on the presence dosage from that one subgenome, but that's definitely not the norm. For most markers and most polyploids, dosage ambiguity is a real challenge.
Well, we have definitely covered a lot of ground today. We journeyed through the evolution of these PCR-based markers, RAPDs, the early, somewhat shaky ones, AFLPs, the more robust but complex ones, and SSRs, the powerful codominant workhorses. We've seen their inner workings, their strengths, and yeah, their limitations like RAPD's reproducibility issues or the sheer labor of AFLP. And we dug into scoring, from the relatively simple diploid case to the frankly complex world of polyploids, highlighting that critical issue of not being able to easily determine allele dosage with these older methods.
Absolutely. These techniques really laid the foundation for so much of modern genetics, especially in plant breeding and conservation. They gave us the tools to finally see genetic variation, track traits, understand diversity in ways that just weren't possible before. They really embody that scientific drive for better, faster, more informative ways to read the blueprint of life.
So what's the takeaway for you listening? Understanding these foundational tools, their power and their pitfalls, really helps appreciate where genetics has come from and where it's going next. And it leaves us with a thought. As technology moves beyond gels towards more direct sequencing, how will our growing ability to precisely count allele copies, even in complex polyploids, reshape agriculture, medicine, and our understanding of evolution itself? Definitely something to think about until our next deep dive.